Okay, so how are the recordings working out for people? This is my first time doing it on YouTube. People can hear me, people can see. Good. Okay. So let's go over some questions from Quiz 4. So get out your letters if you want to participate. Okay, people did really well. There were more 10s than I've ever seen in my life this week. So that means I think people are figuring out how to do it, right? People are realizing you don't need to read 2 chapters and take it, that you can just look up the answers. You can concentrate on certain topics. That's really good. I did get a few email questions, so I'm going to go over those questions now, the ones that people emailed me, and then just some others that people seem to have a hard time with. Basically, if 10% of the class or so gets it wrong, I tend to go over it. Okay, the role of the Golgi in protein maturation is C. Right. So where do A, B, and D happen? In the ER. So that was a dead giveaway. So I think I may have gotten an email from someone about this. Does anyone have a question about this question? Question about a question. Okay. Good. Okay. Macromolecules are delivered to the lysosome by multiple pathways. These include so pretty much there's only one. All of them are going into the cell and one of them is going out of the cell. Right? So you can pretty much say exocytosis does not apply, right? Because exocytosis is going out of the cell, so you would never be able to deliver something to the lysosome when it's leaving the cell because the lysosome is on the inside of the cell. So the traffic's going in the wrong way. I did get one comment about autophagy, and there was confusion over, because the traditional thought about autophagy is that it's digestion of self or self organelles. In fact, it is, but basically think of it as digestion of cell parts that are large, including protein aggregates. So So I used macromolecules. I think that may have confused a few people. Maybe I should have said large things, or not large things, things, or cellular components. No, because then someone might think that's not proteins. But anyways, maybe the wording was a little confusing for some people, so my apologies. Okay. The fate of endocytosed material is lots and lots of B's. I got 1 D. Okay, so for the D people, in order for it to be all, it must be all. So is endocytosed material ever delivered to the proteasome? I mean, maybe there could be one published example of it, but generally, is it delivered to the proteasome? No. Now, this could be confusing for those who are familiar with endocytosis, because often receptors do tend to be monoubiquitinated, and that is a signal for endocytosis, but actually delivery to the lysosome. So, but ubiquitin and proteasome are very related, but then there's mono versus poly. But you guys weren't thinking about that because I didn't teach it to you, and it's not even in the book. Lysosome, that's the right one. Delivery to the vacuole. I did get one email about that being confusing, because the textbook talks about the vacuole being analogous to the lysosome in animal cells, in plant cells. And so I see how that can be confusing. But if you read through that section, you'll see it says that the vacuole is involved in many different functions, such as regulating osmotic pressure, storage of proteins, a whole bunch of things, not just digestion of things, but that is one function. But it doesn't talk about getting it there through endocytosis. And the question was about endocytosis. I apologize if that was misleading. I told this person, is that person here? That was you? So I don't know if you saw your email, but I emailed you back this morning and I told you if you can send me a reference saying that in plant cells endocytosis material is delivered to the vacuole, I will give you the point, and that is for everyone. So if you lost a point because that was a little vague, I apologize. And feel free to send me a reference about endocytosis. I don't know anything about plant cells, so I'm not going to pretend I do. And so definitely send me a reference. It's just not covered in our textbook. Okay. That's it about the quiz, because pretty much everything else, almost everyone got right, which is pretty awesome. Okay, business. Recitation is back on. Those who have not, please bring your Scantrons this week. Exam 1 is coming right up on October 5th. So right now is when you should be going, holy crap, I need to, oh, bad word. Holy, is that a bad word? It is to my kids, right? You don't want like 4-year-olds going around, holy crap. But you should be going, oh my gosh, we should be organizing ourselves. And I'm scared. And so that's good. I want the fear in you now, because now you have plenty of time to get it together. I don't want the fear in you the day before. So yes. I will open up the past quizzes the week before so you can look over. So I've been telling people visiting me in a panic that as you take a quiz, copy and paste everything into a Word document. Keep it, have it, own it, love it. All the practice problems, copy and paste them into another document. And so you'll have, coming up to the exam, plenty of questions to play around with. Don't try to answer those questions, look at the topics. So make sure every topic that's covered. Also recitation questions, because those are very much related. So make sure you've got all your topics covered and think big picture and study with friends. And come to me if you're going through your stuff and you cannot find any pattern and it all seems not connected, because it's all connected and we need to see those connections in order to remember them. Okay? That's my lecture for you now. Okay. Let's move on. The Golgi. So, we were talking about the secretory pathway, and we were talking about how proteins with certain signal sequences enter the secretory pathway because they're destined for secretion, plasma membrane, or lysosome. And so they go into the ER, and what happens in the ER? Folding, basically. So, and the things that need to be happen for folding, disulfide bonds are made, bonds are rotated, big honkin' sugars are added. And then if they fold properly, they're allowed out of the ER, and then they move on to the Golgi, and they enter on the cis phase. So there's a cis Golgi network and then a trans Golgi network. And so they enter the cis Golgi. So if I say CGM, cis Golgi network at this end, so your ER would be up here if we were drawing a picture of the cell, and your nucleus would be up on the ceiling. And then they make their way through and they exit the trans Golgi network towards the plasma membrane or lysosomes, circulatory vesicles, whatever. Okay, just orienting. Now we've got our lay of the land. Okay, and as with all organelles, there's going to be different quantities of them in different cell types based on the job of the cell, right? So an example is a cell that is specialized for secretion, like goblet cells in the intestine that secrete lots of mucus. They're going to have tons of Golgi, right? Probably also have tons of ER, but tons of Golgi, because they are making tons and tons of secretory vesicles full of mucus, right? If there's only a little bit of Golgi, well, that doesn't seem very efficient. So I just always want you guys to keep in mind the fact that some, like which cells have lots of mitochondria? Muscle cells, right? So like cells are not all the same. Different cell types have different amounts of different organelles and different proteins and do different things, right? Okay. Anyways, so what happens in the Golgi? Well, the first thing is, remember those end-linked oligosaccharides that were added in the ER? Well, they are further trimmed and so they're added in the ER, and there's all this trimming that goes on in the ER that's involved with ERAD and whether or not the protein is folded and it's going to bind to the lectin if there's sugars are on it and other sugars are taken off of it, and this and that and back and forth, and finally it gets out of the ER with still some sugars on it, right? A whole bunch of sugars are still on it, and then different enzymes trim and add sugars and change things to basically to end up with a protein glycosylated, how that protein is supposed to be glycosylated. So basically you've got trimming of N-linked oligosaccharides that were added in the ER. You have O-linked glycosylation, so different block of sugars added to the hydroxyl group of serines and threonines. Proteoglycans and sulfation happens. Basically, when you think Golgi, you think addition and modification of sugars. Okay? So who cares? Why would we be adding sugars to proteins? Why are we glycosylating proteins? What's the point? Other than the fact that it's in the textbook. Yes?
I thought it was to aid in the protein folding to make it fold better.
Okay, so there is a role of glycosylation in protein folding. Yep. What else? So where do a lot of these proteins that are in this pathway, where are they going?
The membrane.
The plasma membrane. So a lot of membrane proteins and receptors tend to be glycosylated, right? Okay, so then you're saying, why are they glycosylated? Well, a lot of it has to do with protection from proteases, protection from binding by other proteins. Let's see, what else do I have down here? Right, so they protrude from the protein surface and limit interaction with other proteins, such as proteases. You don't want proteases. Let's say, well, first of all, there's tons of proteases in the cell. So let's say there are proteases, and they can easily get to a protein that you're trying to get to the plasma membrane. You don't want that. If it's on the plasma membrane, let's say it's a protease in the extracellular space coming from, say, a virus or a bacteria. Not the first time I've ever heard of that, right? You don't want that to be broken down. But a big one, which is a favorite of mine, there's a nice movie, which I'll try to put up on Blackboard. Cell adhesion, that's a big one, right? Cell adhesion. What does cell adhesion mean? Just put the words together. Yes. Cells sticking together. Why would cells need to stick together? Huh? Maybe for communication. Or signaling. Yep. Well, I have an example, right? So the example, and there's a movie that I will put up. Someone remind me if I don't. Selectins. These are receptors, and they have lectin domains. What are lectins? What do they bind to? Selectin. Well, lectins bind to sugars. So you should always, if I say lectin, you say sugar. If I say tomato, you say tomato. No. Lectins bind to sugars. Okay, so the story is selectins are receptors, and they have this domain. When I say domain, it's just a business end of a protein, right? Proteins, you can think of them as machines with different parts that do different things. So one domain is a lectin domain, and it will bind to sugars. And so this receptor will bind to the sugars that are on endothelial cells. So why would you need to do that? So here's your blood vessel, right? Right? Okay, is everyone thinking blood vessel? Think of it as a cross section, right? So you've got endothelial cells lining your veins or whatever you want to call it, right? And then you've got, what goes in between those cells? Let's say you have a vein. What's inside of it? Blood. Come on, guys. So blood, plasma, things containing cells, right? What's in blood? Plasma. What's in that? Cells, right? Come on, people. Is this Bio 101? Do they have a 101 here? No. Why not? Shouldn't the intro always be 101? Or they want to boost you up 201, 201. Okay. For me, it was Bio 110. I always wondered where 101 through 109 went. Okay. So you've got cells in the blood, right? Some of those cells, white blood cells, they fight infection, right? Okay. So I got you. You're in your blood vessel. You're a cell, right? Okay. So let's say like your skin is up here and you get stabbed by an angry classmate who didn't get an A. That'd be me. I was stabbed by the angry student who didn't get an A. There's an infection developing at the site of the stabbing with the lead pencil. Remember your teachers always be like, be careful, you don't want to. Monday mornings are hard. Are Monday mornings hard? Yeah, okay, good. Just making sure. So what needs to happen, right? These white blood cells that are in your blood need to get to the site of infection, right? Right? Otherwise, what's the point if they're just in the blood and you've got like this festering infected wound on your skin, right? So they need to get to the site of infection, and that means they need to get out. Did you ever wonder how the white blood cells get out of the blood to the site of infection? Did you ever wonder? Do you guys sit up late at night thinking about this? I bet all of you right now are going, oh my God, I never thought about that. Well, this is how it happens, okay? So your white blood cells, they have these selectins, so they've got these receptors with this lectin domain that binds sugars, and they actually bind to sugars that are on endothelial cells. So think about how fast your blood is moving in your blood vessels, right? Pretty fast. Anyone have any miles per hour for me? So, 20 miles per hour. I think it's faster. It's, I don't know, 120? I don't know. Someone's just guessing now. So they're going, right? And then what happens is immune response, gene expression, proteins made, receptor goes to cell surface, right? Selectin is now on the cell surface, and think of it like Velcro, right? It's like the little hook part of the Velcro, and it sees on the endothelial cells all these glycosylated proteins that are on the surface of the endothelial cell, and it binds to it. And there's tons of these selectins all covering the immune cell, and they bind to the endothelial cell, and they slow down, and they're rolling, and then they stop because they're bound. Many selectins are bound to many glycosylated proteins on the endothelial cell. And then other things happen that cause the spaces in between the cells, the endothelial cells in the blood vessel, to kind of open up a little bit, and you have seeping of fluid through, and the cells get through and get to the stab wound. By now, campus police has come. And that's why when you have inflammation, when you get hurt, it gets red, it gets hot, it gets puffy, right? Puffy for all that fluid. Anyways, so here's an example of why glycosylation is important, right? Just one example. That's not the only reason for glycosylation. But so you have glycosylation of proteins that end up on the endothelial cells, and then it is involved in stopping white blood cells and stopping them and allowing them to go through the blood vessels. Okay, does everyone enjoy that? Okay, good. Okay, let's move on. So, the ER has all these different compartments, right? I said something about there's a cis Golgi network, and then there's a trans Golgi network. And so what happens is you've got your vesicle containing your protein, and it goes to the cis Golgi network, and you start to have movement through all these different compartments, and in each compartment there are different enzymes and different functions. Different things happen in different parts of the Golgi. And then eventually it comes out the trans Golgi network and goes wherever else it needs to go. Yes? Will every protein move through every compartment?
It doesn't need some of these things. Will it travel through there anyway?
You know, I don't know. That's a good question. So does every protein need to go through every compartment if it doesn't need some of those things? My guess... Oh, okay. Oh, yeah, I'm sorry. Sorry about that. Actually, I put up a huge extra credit if anyone wants to make a movie. That was like 20 points you guys can get. No one's biting. Huh?
I feel like no one has free time.
I know, no one has free time. Who's a film major? Who was a film major? Did you know I was a film major? See what can happen? Who wants to be a film major? Because you can change. Anyways, extra credit. Let's do an extra credit. Thank you. Thank you, troublemaker classmate. Okay, so let's do an extra credit. The question is, and you guys have, that means you have 24 hours. So you have until 11:30 tomorrow morning to email me a referenced answer. So emailing me something about what you think is not getting you anything. You need to have a referenced answer where I can open up the file or look at the link or whatever and see exactly where, like, that what you're saying is supported by real data. Okay, so does every protein need to go through every compartment of the Golgi? Because what if, for example, Glucknack does not need to be added to that protein? Does it need to go through there? I have my thoughts about the answer, but we'll wait for the data to come in. Yes. Yes.
I thought it was compartment maturation. So.
Well, right. Well, so we're going to get to that, but you know what? We're going to get to that. So I know exactly what you're heading towards. And it might be even, it's right there. Okay. So yeah, we're going to talk about that, but still we'll make it an extra credit because I haven't done any yet, other than 20 points for filmmakers. Okay. So this is just how they actually studied the fact that there are different enzymes in different compartments. This is an EM. These are EM pictures, it's straight out of the textbook, where they stain for different enzymes. So let's turn the light on. So see these are Golgi. They look like these stacks right there, right? And so you can see here's unstained, so nothing stained. And then they stain with different types of enzyme. They don't stain the enzymes, but they usually put a substrate in for an enzyme that then they can stain that substrate, or they can use an antibody against an enzyme. And you can see here, this is your. Let's see, this would be your. This one. I forget which way is going which, but it looks like ER and then out. So you would have one kind of enzyme here. I don't have them labeled. It's in the textbook. Different enzymes are here. Different enzymes are here. So this would be trans, and then the middle, and then cis. Basically, that's how you study things, right? You can look at them. You can do microscopy and stain for different enzymes. Okay, so this is what we were just talking about, right? Two models. So there are two models for transport through the Golgi. And so your question, depending on which model you believe in, or whether it in fact is a combination of both, may or may not be relevant. But that's why it's because there's two models means it's not really known, which means that this might be a good extra credit. But there are two ways, right, that you can transport through the Golgi. One is this vesicular transport, which makes your question very viable, right? Because you've got vesicles coming from the ER, turning into these vesicular tubular clusters, and then going in and then coming out and kind of bouncing from section to section. And so I could see not needing to go somewhere. I doubt it, but I could see it. But then there's this cisternal maturation model, which is more about it's more about the contents of the Golgi moving towards the vesicular tubular cluster than the vesicles actually going to each section. So you've got these you've got these vesicles that become the vesicular tubular cluster, and then this cluster becomes the cis, which becomes this section, which becomes this section. So those enzymes are in contact with each cluster, which is going to contain many different proteins. And so that would suggest that that doesn't happen, that actually everything goes through every. So it depends on which model you believe. So I look forward to everyone's answers for this question. And some people believe that it's a combination of the two of these, which then goes back to your question. Well, maybe if your protein doesn't need to be, what's the word? Treated by a certain enzyme, let's say, maybe it'll skip over something. I don't know. The cell does like to do things in an efficient way. So in a weird way, cisternal maturation, which is this model, makes no sense because it doesn't seem very effective. But who knows? So I look forward to everyone's thoughts. Okay, let's go to the lysosome. So the lysosome is a site for ubiquitin-independent degradation or digestion. Independent. So the way it works is it contains acid hydrolases that are only active. So these are enzymes that degrade things that are only active at a very acidic pH. So the pH in a cytosol is 7.2, and the pH in the lysosome is 5. And there are all these different hydrolases, nucleases, proteases. What do nucleases degrade? Nucleic acid. What do proteases degrade? Just making sure everyone knows their aces. Glycosidases, sugar. Lipases, lipid. Phosphatases, phosphate groups, right? Sulfatases, sulfur groups. And phospholipases, phospholipids. Okay, so pretty much anything can be degraded, right? If you put a whole vesicle, like all the lipid from a vesicle can be degraded, right? Okay. Someone's ringing. Isn't it so funny? It's vibrate, so you're being very polite because you're on vibrate, but I could totally hear it. Okay, so what am I saying? Right, we're talking about pH. Oh, right. Okay, so contains all these acid hydrolases. They're only active at an acid pH. So how does this protect the cytosol? So what I'm saying is, if it's only active at a acidic pH, how is this a protective measure that the cell takes? Right, so you don't want to break everything down your cell. Let's say something goes wrong, and an acid hydrolase. Okay, so the whole point of me bringing up acid hydrolases is that they're in the lysosome. They got to get to the lysosome, right? Which means they start in the ER. They go ER Golgi to lysosome. Okay, that's where we're coming from here. So let's say something goes wrong, and their protein is translated into ER, modified to the Golgi, and somehow something goes wrong, and it goes to the wrong place. It goes to the plasma membrane, right? The thing that's supposed to direct it to the lysosome, it doesn't work, doesn't happen, and it goes to the plasma membrane. What's going to happen? Plasma membrane is, let's say it's a phospholipase. What's a plasma membrane made of? Phospholipid, right? Yeah. What were you going to say?
I was going to say that the phospholipase wouldn't be effective at the pH of 7.2 in the phospholipase.
Exactly. The phospholipase wouldn't be effective at all, right? It wouldn't be active at the pH of 7.2, which is what the cytosol is. So who cares if it goes to the wrong place? You're fine. It's not going to degrade your plasma membrane. So that's part of so it's kind of a level of protection for the cell that they're only active in a highly acidic environment, which would be the lysosome. And the other thing is, so these proteins are made in the ER and the Golgi, right? So at the end of the ER, it's folded properly. It goes to the Golgi. Why isn't the Golgi and all the things inside the Golgi destroyed, right? Same reason, right? The proteins are not active yet because the pH is not 5. So there you go. Questions? Does anyone want to know why I'm talking about this? Is anyone wondering what this has to do with the secretory pathway? Someone? So everyone's on board. Okay. I see some nods. I see some people averting eye contact. Okay. So notice down here we've got a proton pump, right? So that proton pump is pumping protons in. What is the purpose of a proton pump in this case? pH, right? It's lots of protons, make it acidic or basic? Acidic, right. So that's how your pH is maintained. And then remember we were talking about chloroquine the other day. Chloroquine, I believe, does something which prevents the proton pump from working. So it prevents lysosome acidification. Okay, good? Good. Okay, so this is what lysosomes look like by ER. And they actually were staining acid phosphatase by incubating phosphatase substrate in the presence of lead. And so the lead it's EM, so it's not like fluorescence. So the way the staining is a little bit different. So that's why they use a lot of heavy metals a lot of the time. So you could see lots of acid phosphatase in here. See the black? Questions? Okay. So this is the merging of the secretory pathway with the endocytic pathway, right? Because we have things that go to the lysosome, but then we also have the endocytic pathway, which is things outside the cell being endocytosed and going to the lysosome, right? So they become endosomes that then become lysosomes. So things that go to the lysosome, those are mostly the acid hydrolases, the proteins that work in the lysosome when we're coming from ER to Golgi to lysosome. And then you've got this lysosome that's super acidic and full of all these enzymes that's ready to meet an endosome, which will contain something that's endocytosed. Okay, so things can get to the lysosome through different ways, as you all saw in the quiz. So this is kind of just a very basic picture showing this, straight from the textbook, right? You can have endocytosis, you can have phagocytosis, you can have autophagy, and then they all somehow either fuse with or mature into a lysosome. Okay, so now that we know what the lysosome is and what its function is, we need to think about these lysosomal proteins, these acid hydrolases. How do they get from the Golgi to the lysosome? Yes?
Sorry, this might be a stupid question, but if something is going like to the lysosome for digestion, does it go straight from the ER? It doesn't go through both.
It doesn't go anywhere near the ER or Golgi. So yes, so that's why I was trying to point out, okay, so you got your cell, nucleus, ER, Golgi, right? Okay, is that nice? N, E, G. I know, Golgi's supposed to be a lot more detailed than that. So when you're talking about ER and Golgi, we're talking about making proteins that are going to go somewhere to do something, the secretory pathway. When we're talking about the endocytic pathway, we're talking about sending the trash to the lysosome from elsewhere. And so with endocytosis, you're going to have endocytosis of whatever, and then that's going to go, where's our lysosome? One lysosome, one per cell. And that's going to go directly there. So here you have all your acid hydrolases that are going ER, Golgi, lysosome. Okay, did anyone else have that question? Does anyone need clarification on that question? Okay, good. I don't know what I drew up there, but hopefully I got a point across. Okay, So how do lysosomal proteins get from the Golgi? So now forget about the endocytic pathway for a minute. We'll come back to it. We're going back to making proteins that need to go and work in the lysosome, such as acid hydrolases. So how do they get there? Does anyone remember from their reading? There was something, right? Because why don't proteins that are supposed to be secreted go to the lysosome? Oh, yeah.
The mannose-6-phosphate.
I'm hearing, yeah. So I'm hearing a lot of six. Mannose-6-phosphate. Right, mannose-6-phosphate. What is mannose-6-phosphate? I didn't even tell you. See, this is why I make you read on the weekend, because you can come in here and go, oh, right, mannose-6-phosphate.
Signal patch.
Well, it's attached to a signal patch. So mannose-6-phosphate. Mannose is a what? A sugar 6-phosphate with a phosphate group. Added to the protein and a signal patch is recognized. So we'll see what it looks like. Right. Questions so far about what we're talking about? Good. Okay. So they're tagged. You can think of things being tagged with mannose-6-phosphate. So here's your lysosomal hydrolase, and it's got its N-linked oligosaccharide and mannose-6-phosphate. So there's your mannose-6-phosphate, however that looks. There's your phosphate. It's added. It's tagged right on to the acid hydrolase. Why are they tagged with mannose-6-phosphate? I'm telling you they're tagged. I'm telling you that's specifically for acid hydrolases destined for the lysosome. Why? Yes. To be recognized by the M6P receptor. So are you guys noticing a trend in cell biology? That specificity is all about things and receptors binding to each other. Things and receptors. That's the scientific terminology. Things and receptors. Right. So, exactly. So there's going to be a receptor that we're going to see soon. Right. Okay. So where is the mannose-6-phosphate receptor located? Where do you think it would be? Like, in what part of what organelle?
I might have read it wrong, but on the end side of this. I don't remember.
Okay, it's okay. I hear you saying. The membrane of the lysosome. Well, where does the membrane of the lysosome originate? Yes.
I was going to say that in the trans-Golgi network.
Yes, in the TGN trans-Golgi network on the membrane, right? So remember, right, we've got ER, Golgi to lysosome, right? And here's your acid hydrolase with M6P, right? And so you're going to have something there that binds to it, right? And then it's going to bud out, and you're still going to have it in there, right? And then it's going to fuse here. And so you will end up, sorry, that's not a good spot. So you will end up with your mannose-6-phosphate receptor, right, because it's going to fuse. Remember, we're having fusion, maintaining topology. You are going to end up with your delivery of your acid hydrolase with the mannose-6-phosphate and the mannose-6-phosphate receptor to the lysosome, right? So then there's going to be a whole other issue of, well, you really don't want your mannose-6-phosphate receptors all ending up in your lysosome because they got to go back. Yeah. What are we talking about? I don't know. Yeah, there's going to be more figures. So we're going to keep going, and we're going to answer your question. Yes.
Well, what actually drives the acid hydrolases there? Is it like a motor protein?
Well, so all the movement of these vesicles is motor proteins on cytoskeletal proteins. Yes. So the question is, what's driving it there? Yeah, and that's exactly. So you're going to have vesicles that are budding and going different places. You have vesicles going from the ER to the Golgi. You have vesicles going from the Golgi to the plasma membrane. You've got vesicles going from the Golgi to the lysosome, and they all are driven by microtubules and motor proteins. But the question is what's going in what vesicle and where is it going? And that's the Rab proteins and the snares and the And then these receptors that select certain cargo that's going certain places. Okay, so we're gonna move forward with your stuff, and you're gonna tell me afterwards if you get it. Okay, so how does the Golgi? So first, how does the Golgi even know to add mannose-6-phosphate? Right? Because remember, mannose-6-phosphate is a tag that's added to proteins going to the lysosome. So, so how does it distinguish? Right? Because all proteins are leaving the ER with all the same sugars. And then there's all this trimming going on in the Golgi, in addition of sugars, including mannose-6-phosphate. Well, there's a signal patch that was brought up earlier that is on this acid hydrolase, and it's recognized by glucanac phosphotransferase, whatever, enzyme that will add mannose-6-phosphate. What is a signal patch? We've talked about signal sequences. What's a signal patch?
Isn't it the signals found in the tertiary structure of a protein?
A signal found in the tertiary structure of a protein. Did you want to add to that? It's pretty much the same. Anyone want to add? So what do I mean by what does he mean by tertiary structure of a protein? Yes. Right. So the surface of the folded protein, right? Yes.
So is it kind of like an active site, basically?
Right. Well, so you can say it's comparable to the concept of an active site being made up of different domains of a protein when they're folded together.
There's no conformational chain associated with it.
Well, and not all active sites. That does complicate things. But so an active site usually has some kind of catalytic activity, whereas a signal patch is nothing but a surface that's made up from different domains that come together when a protein folds. And so they're showing it here, right? So here's your acid hydrolase, and it has this, see how it's all folded? It's a protein that's folded, and all this whole bottom part is the signal patch. And so you'll notice it's made up of a very, N-terminal, some mid protein, and some C-terminal regions. But they're close in space, and they make the same surface when they're folded together. Kind of like, if you crumble up Play-Doh, then you'll have something, but then if you roll it back out into a long string, maybe that same part will be different places, right? So what happens is the signal patch is recognized by this enzyme that's going to add the mannose-6-phosphate. So the signal patch binds, and then mannose-6-phosphate is added to the N-linked oligosaccharide that was added in the ER, right? And then then it's released, basically. Signal patch. Okay, so we're still getting to your thing. Okay, I didn't forget you. Okay, questions about this? Yes.
Does the M6P bind to the signal patch?
No, so the signal patch is recognized by the enzyme that adds the mannose-6-phosphate. So here's where if you can see here is where the signal patch is recognized. So it's holding on to the protein, and then there's this catalytic site of the phosphotransferase that's binding mannose-6-phosphate and adding it. So it basically, the whole point of the enzyme is to hold on to the protein and position it in the right orientation so that the sugar could be added correctly. Okay? Questions about this? Okay. Okay, we're good. Okay, so lysosomal storage disease. This is just a case study for you guys to think about it. What happens? Okay, so there's a 3-year-old who presents with multiple musculoskeletal abnormalities, and you, the brilliant doctor that you are, suspect eye cell disease, which is a severe lysosomal storage disease. You find low lysosomal activity when you look at multiple cells. So you take a bunch of cells from her blood and wherever, and you test for lysosomal enzyme activity, right? Because you know the enzymes that live in the lysosome, and you check to see if they're active, and you detect all of the lysosomal hydrolases. They're all in the blood and in the urine. So there's in the actual lysosome, there's no activity of these enzymes, but you find all the hydrolases in the blood and urine. why might this be? So how come there's no activity of the acid hydrolases in the lysosome, but you're seeing it in blood and urine? So stop and chat for just a minute, and I'm going to make sure I hit your question. Yep.
I'm thinking what's happening is it's in the blood, which means that the tomato is being added. Getting giant chicken and the egg problems in my head. Like how do they realize it? Like where did all this come from?
Okay, guys. In the interest of time, I know I hate to break up a rousing discussion, but in the interest of time, who can tell me what they think might be wrong, which is causing no lysosomal hydrolase activity in the lysosome, but lots of lysosomal enzymes in the urine and blood? Anyone want to give me a yes?
They're being secreted
They're being secreted. Why are they being secreted?
because the mono-6-phosphate isn't being added.
Mannose-6-phosphate isn't being added. Why might the mannose-6-phosphate not be added?
The enzyme that's supposed to recognize the signal patch that add M6 is probably defective.
So the enzyme is defective. Why isn't the signal patch defective? Why enzyme and not signal patch? See, now I'm doing to you what they do during oral exams in grad school. Every time you have a right answer, they're going to ask you another. Your answer is right, by the way. Yes.
In single patch studies, you're actually getting a protein, but it's not going.
Wait, say what you're saying?
Well, the proteins are still being made,
Right.
so your single patch is probably fine, but you're still getting them, they're just not going.
Signal patch is fine, but the proteins will, okay. So, but the signal patch is what determines, is what binds to the enzyme that adds it, right? So, did I have someone over there?
I was just gonna say, the signal patch wasn't there, then the protein was probably not folded right, so
Right, okay, so that's kind of what you were saying, right? So, okay, so I'm not hearing exactly what I want to hear yet. Yep.
Couldn't it be a number of things with anything from like just a wrong protein folding to a defective enzyme that adds the mono-6-phosphate or some kind of genetic deformity?
So the question is it the enzyme or is it a protein issue? Is it an enzyme issue or is it an acid hydrolase issue?
It seems like it would be the enzyme itself because that only takes one thing to go wrong.
Right, okay, only one thing could go wrong with the enzyme and that could cause a whole big mess. But with proteins, it's going to be a little different. Every protein's different. Yep.
Could it possibly be a silent mutation?
A silent mutation. What would a silent mutation do?
It's as opposed to like the signal that would be the signal patch, something in the signal patch would be changed, so the enzyme would be fine, but it could recognize the signal patch would be.
Okay, so we're kind of going back and forth. In the interest of time, I'm going to tell you guys. Was there another comment over here? Not quite. That's what I want to tell you.
Yes, it could be a defect in the receptor protein on the lysosome.
Oh, it could be a defect in the mannose-6-phosphate receptor. Right? Okay. So when you ponder these questions, you want to think, well, what's going to affect something so huge, right? So it's got to be the enzyme or the receptor. Would it be the signal patch? Well, every acid hydrolase is going to have, every protein has a signal patch. So that would mean you'd need like 10 mutations, right? Because you're not finding anything in the lysosome. So it's got to be the enzyme or the receptor. I am pretty much out of time, but this is the mannose-6-phosphate receptor slide that should explain all of your questions, but we will start with it on Friday.